Hello, everyone, and welcome to today's webinar, Precision Oncology Unleashed, Challenges and Innovations in ctDNA and Deep Sequencing. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Burning Rock DX. To learn more, visit us.brbiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers for today's webinar. Dr. Xinru Mao, Head of Pharma Services and Global IVD Business at Burning Rock DX, and Dr. Bingsi, Vice President, Head of Innovation Center at Burning Rock DX. Our first speaker is Xinru Mao. She is responsible for leading the pharma biomarker at CDX Services, including business development, translation medicine, and project operations globally. She also leads the IVD international business, including go-to-market strategy, commercialization of arrays, and channel partner development and support within the regions for Americas, Europe, Japan, Middle East, etc. Dr. Xinru Mao has over 16 years experience of innovative drug clinical development and NGS-driven precision medicine in oncology therapy. Dr. Mao has been with BNR since 2015 and has held several positions with the company. During the 5 years prior current roles, she served as medical director who led the medical affairs team. Dr. Mao, you may now begin your presentation.
Okay, thanks. Hi, everyone. I'm Xinru Mao. I'm happy to be share my topic about ctDNA sequencing in cancer research and clinical application. I will share some ctDNA detection highlights and challenges, and also highlight some clinical utilities of ctDNA application in the oncology, and also disease monitoring with a series of ctDNA. So this is my disclosure. So firstly, let me introduce who we are. Burning Rock, specialized in NGS diagnosis solution for precision oncology, which listed in NASDAQ in 2020. We have 3 business units. The first one is the therapy selection for cancer patients. We deliver this business with 2 models, the central lab model and the decentralized model. Up to now, we had tested more than 750,000 tests with our central lab, and also partner with more than 100 labs with our decentralized solution. We provide the biomarker and CDX service for the pharmaceutical companies. Currently, we work with more than 500 clinical trials and 15 CDX co-development projects with more than 100 pharmaceutical partners, such as AstraZeneca, Johnson & Johnson, Lilly, and so on. Also, we have the early detection section, which is provide the multi-cancer detection for these high-risk individuals. This also received the FDA breakthrough designation and the CE mark. Besides this, we published more than 600 articles, and we have 2 labs in the US and China, which certified by the CAP and CLIA. We also have the ISO 13485 and 15189. We have more than 1,000 employees. So Burning Rock provide end-to-end oncology solutions. Our comprehensive genomic profiling can provide the testing for both solid tumors and hematological malignancies, which includes the small, middle, and large signs targeted panel, both for the tissue and ctDNA testing. And also we have WES, captured WBTS, and WGS. For the hematological malignancies, we have DNA and RNA dual biopsy assays for the testing. And our can-catch NRD series solutions, which can provide the NRD detection, including the tumor-informed personalized NRD-assay and tumor-naive methylation-based NRD and tumor-naive epigenomics and genomics dual liquid biopsy. So the early detection panel is can provide the multi-cancer detection with the blood test. So today's section is focused on the OncoCompass comprehensive genomic profiling with the ctDNA samples. Let's step to the NGS-based ctDNA test in cancer research and clinical practice. As today's topic is about the ctDNA, let me introduce some background about the ctDNA biology. So it is released mainly through the apoptosis, and the fragments carries characteristics of the tissue of the origin. In addition, it have very distinguished fragment size due to the way to attach to the nucleosomes. Moreover, it cleared very quickly in the circulation by the liver and by kidneys. It has very short half-life of minutes to hours, so it is a highly dynamic biomarker. In normal practice, cfDNA shedding depends on the tissue cell turnover, so it is not a surprise that the majority of the cell-free DNA comes from the hematopoietic cells in the lung cancer. Finally, the ctDNA shedding is affected by multiple factors. The most important one should be the disease burden, and there are many other tumor biological features, including the tumor proliferation, apoptosis rate, and other factors may impact the amount of ctDNA released from each site. So the ctDNA is transforming the landscape of the oncology drug development, offering a non-invasive, highly informative tool to support the decision-making across multiple stages of the clinical pipelines, starting from the exploratory study to the therapy selection and the efficacy analysis. The ctDNA methylation can be used for NRD detection to optimize the treatment. So as we know, the high ctDNA and tissue concordance can enable the confident clinical trial decision. We do the consistency analysis of the real-world data from clinical testing by our OncoCompass standard, with around 1,000 paired samples of lung cancer. The study results showed that in the all variant types, we can achieve high PPA and sensitivity of 93.4% and 76.8%. For the SMP and indel and fusion variants, it is also have the high PPA and sensitivity. Although calling the copy number variation and large genomic rearrangement is quite challenging, we also achieved the PPV as 70.9%. When we boost to the therapeutic targetable variants, it is also showed high PPN and sensitivity, which support the treatment decision making. We listed the details in this table about the target variants. So OncoCompass also can be used to detect the ESR1 mutation in breast cancer. As we know, up to 40% of the endocrine therapy resistance cases are driven by the activating mutation in the ESR1 ligand by binding domain. Burning rock ctDNA assay can detect a broad spectrum of EGFR mutation. This graph shows the profiling of the common ESR1 alteration in the metastatic breast cancer. We do the accuracy study, compared the OncoCompass and the original method in detection of the ESR1 mutation. In the total 3 batches of OncoCompass panel was used to test to the reference samples and the contrived samples. The results showed very good with high with 100% PPA and NPA. In terms of the patient outcome, in the FRIEND study, the results showed that the patient received the fulvestrant showed a trend toward improved PFS in this ESR1 mutant patient detected by the OncoCompass assay. So our NTAP loss, our OncoCompass ctDNA panel also can cause the NTAP loss, including the NTAP whole genome hemorrhagosis loss and the exon level loss. The scarce lot, these two scale loss showed an example of this kind of two variants. So in this report, it showed understand the co-mutation of the NTAP loss is essential for the design of the therapeutic strategies, especially for this therapy leveraging the synthetic lethality for the approach to the targeting the PNNT5. This graph showed the landscape of the co-mutation of NTAP loss. This is a phase 2 study which used OncoCompass as the therapy selection and the mutation. This study used an anti-EGFR monoclonal antibody to treat the refractory RAS-RAF wild-type metastatic colorectal cancer. It is published in the eBiomedicine. This patient was selected as the KRAS, NRAS, BRAF negative in the ctDNA by Burning Rock OncoCompass ctDNA assay. The median PFS is reached 5.1 months, and the median OS can reach 16.2 months. So it is comparable to the reported data. And the study also showed the ctDNA clearance status have the better PFS and OS. It is a case to use the ctDNA panel to identify the mechanisms of the acquired resistance, which published in the Cancer Cell. It is a phase 3 trial to compare the olatinib monotherapy or plus or monotherapy or the combination to extend the PFS of the patients. The blood sample was collected at the baseline and the disease progression. It is found that at the baseline, the mutation, the molecular mutation landscape was comparable between the two therapy groups. At the disease progression, 57% of patients developed non-resistant mechanism. When we compared the acquired resistance between the two therapy groups, it is found that the combination group had the lower EGFR T790 mutation and the high unknown resistance mechanism. This is the first to report that increased blood-based intratumor heterogeneity is associated with unfavorable outcomes, immune checkpoint plus the chemotherapy in advanced non-small cell lung cancer, which published in the BNC medicine. The BITH score was calculated by the allele frequency of the somatic mutation detected by the OncoCompass. This study divided the patients with two groups, the BITH score up and stable or down. It is found the BITH up group was defined as 10% increase compared to the baseline. The study result found that this patient who have the BITH score up had the shorter PFS and lower response rate and the less mechanism tumor signs decrease. It is a case to indicate that the BITH score can be a predictive biomarker. The blood sample was sequenced at the baseline, the first follow-up, and the disease progression. Although the patient disease remains stable at the first follow-up, it is found that the BITH score was sharply increased with a lot of loss of function mutation occur. These mutations are still maintained, presence at the disease progression with a higher allele frequency. Moreover, a lot of other new mutations come out, such as the PI3K and the PTEN. Another study is used the ctDNA to make a stratification strategy. It is a multi-center randomized placebo-controlled phase 3 trial called Choice 1 on the first-line chemotherapy plus the PD-1 inhibitor for patients with non-small cell lung cancer. A series of the blood sample was prospectively collected in this study from the baseline to the on-treatment and the post-PD time point. This plasma sample was detected with the OncoCompass standard version and low-pass WGS. And then the study performed the analysis of the pre-treatment ctDNA analysis and a blood-based genomic immune subtype. It is defined as BGRS score. By evaluating the status of BTMB, BITH, and CRM. Also, it performed the ctDNA analysis on the treatment of and PD. This study aimed to establish the ctDNA-based stratification strategy for the immunotherapy in patients with non-small cell lung cancer, and also independently validated based on 2 phase 3 trials with the same design. Finally, the study recommended a therapeutic algorithm based on the BGI score subclassification. The study found that the patient with the baseline ctDNA negative, it is a BGI score, BGIS group 1. It should be a low ctDNA shedding group. In this group, this patient who received the first chemoimmunotherapy, first-line chemoimmunotherapy have the beta PFS, but the OS no differences. So it is indicated that this group have the limited long-term benefit from the first-line chemoimmunotherapy. And when the baseline ctDNA is positive, if the patient with BTMB high or BITH low or BCRN low, it is BGIS group 2. In the group 2, it is found that the patient have better PFS and OS while received the chemoimmunotherapy. This group should can strong benefit from the first-line chemoimmunotherapy. And the group 3 is the BTMB low with the BTMB low and BITH high and BCRN high. This group with no difference between the PFS and OS when they received the chemoimmunotherapy. So this group should be limited benefit for this therapy. So and also besides the blood sample ctDNA testing, we also can provide the testing for CSF and urinary. I will use 2 slides to introduce this scenario. So as we know, the plasma ctDNA have limited utility for the CNS metastasis. In this case, compared to the plasma sample, the CSF should have an accurate reflection of the intracranial tumor biology. This study involved in 584 patients, non-small cell lung cancer patients with CNS metastasis. The study results showed that the patients who detected ctDNA in the CSF have the high tumor burden and also associated with significant worse survival. And this patient who received matched therapy with the CSF guided was showed significantly improved survival. We also test the study also tests the CSF ctDNA the disease progression. So this table showed the resistance mechanism detected in this sample. And the study also showed this patient who received a second CSF guided target treatment have a better OS. So in conclusion, the CSF ctDNA should be a variable tool to test this patient with CNS metastasis. And OncoCompass should be a good panel used for the CSF testing. When we step to the urinary ctDNA assay, it is assay designed for a sensitive detection and efficacy monitoring in blood cancer in the urinary cfDNA profiling. The sample, it is required 40 minute sample size per patient, and ufDNA import is ranking from 20 to 60 nanogram. The accuracy, the analytical validation result of the LD showed is around 0.3%, demanding a strong sensitivity in detecting the low-frequency SMV, indel, and fusion. The LB study summary in this table is indicated that the per-site false positive rate was 0 for the actionable variants, including the FGFR alteration and ERBB2 amplification. So let's go to the challenges and perspectives of the ctDNA detection. So as the first slide, we see the biology of the ctDNA. It is indicated that the ctDNA fraction could be the highly depending on the tumor burden. So it is so in the later stage cancer patients, the ctDNA abundance is around 0.2% to 0.5% in most cases. While for the early stage cancer patients, the ctDNA abundance is quite low, it's less than 0.01%. For example, for this MRD detection scenarios. So it is quite important for the sensitivity of the ctDNA detection. Due to the ultra low of the signal abundance, the noise in detecting is also very important. So the specificity should be another challenge to achieve an effective detection. And there are two kinds of the noise, the technical noise and the biological noise. The technical noise can come from the experimental error and post-processing error, such as sample preparation or DNA degradation or bioinformatics analysis. And the biological noise can be found in the cfDNA, which is not originating from the tumor cells. such as clonal hematopoiesis mutation and the age or non-tumor related DNA methylation. So to address this issue, several strategies is developed to improve the ctDNA sensitivity and specificity. To improve the conversation, can boost high sensitivity of the ctDNA detection by enhancing the ligation, more DNA import, or sequence to saturation. Reduce the technical and biological noise can increase the specificity by using the unique correction or phased sequencing, or use the WBC paired sequencing to reduce the chip mutations. And increased size can improve the sensitivity for such as use the whole genome approach for the NRD detection. So we take consideration of the different scenarios of ctDNA use for the clinical practice or the pharma drug development. We develop the standard ctDNA assay and ultra-deep ctDNA assay. The standard version is tested with sequence deeps at 10,000 with the LD close to the 0.4%. The ultra deep version was sequence deepest was 20,000 to 50,000, and the LD came down to 0.07% at 50 nanogram. So so it should be the last slide on my side, and I will introduce the Dr. Bingsi. She will give more details about the ultra deep ctDNA assay. Thanks for your attention.
Thank you, Dr. Mao, for that presentation. We do have a couple of questions regarding your presentation. Our first question asks, given the advancements in ctDNA testing over the years, particularly the improvements in sensitivity you mentioned, which highlights its capability in detecting rare variants, how do you foresee the trend of ctDNA being used as an alternative to tissue testing in clinical trials?
So I think the growing sensitivity and specificity of the ctDNA testing are promised for the role of the ctDNA used for the clinical trial, which may towards partly or fully replacement of the tissue in the future for these scenarios, such as these cancer types or this kind of the tissue sample can get difficult. And as we know, and also as we know, the sample is limited. So I think in the future, the tissue biopsy reserved for this case with undetectable ctDNA or ambiguous result. In additional, ctDNA response monitoring and use ctDNA to monitor response and NRD application can be a series of scenarios to replace the tissue sample for during the treatment for the clinical practice use.
Wonderful, thank you. Our next question that we have here asks, what challenges that ctDNA detection could have in low shedding cancers, and are there any corresponding solutions?
Yeah, I think in the slide there's some information about that. The ctDNA detection is a challenger, especially in these cancer types with the low shedding, such as the CNS tumor or this cancer with CNS metastasis, the breast cancer or pancreatic cancer, and these cancers with the early stage. So for these cancers, some strategies can be addressed for the ctDNA detection. As just mentioned, the unique correction or ultra-deep sequencing, or maybe some other methods such as we use multi-omic approach, such as we use methylation analysis or fragmentomics, or use protein biomarkers. Also, we can also see so for this patient with the brain tumors or brain or CNS metastasis, can use CSF to detect.
Wonderful.
Yeah.
Okay. And our last question that we have here asks, how might ultra-deep ctDNA change clinical pharmacology or translational science?
I think it's a good question that monitor ctDNA dynamic in real time. It should can identify the biological effective faster than the relying on imaging or toxicity alone. So it may be used in the future clinical trials, and also use ctDNA as a dynamic biomarker to track the treatment escalation. For example, the NRD guided the de-escalation or de-escalation therapies for the ctDNA negative patients. Also, in addition, so to accelerate the drug approval, the ctDNA maybe can be a surrogate endpoint for PFS or OS in the future. That's all.
Wonderful. Thank you, Dr. Mao, for that wonderful presentation. I'd like to now bring in our second speaker, Bingsi Li. Dr. Bingsi Li brings over 15 years of expertise to the field of genomics research and the development of next-generation sequencing technologies. She holds a Ph.D. in molecular biology and genetics from Cornell University and a B.S. in medical sciences from Peking University. At Burning Rock DX, she leads the Innovation Center, where she and her team have spearheaded the development and launch of several pioneering products. These include tissue DNA and RNA dual profiling solutions, OncoScreen-made, OncoScreen Capture WTS, blood-based ultra-deep mutation sequencing processes or products like OncoCompass, Target Prime, OncoCompass Plus Prime, and a multi-cancer detection assay, OverC. Currently, her team is developing tissue-free minimal residual disease, or MRD tests, set to launch in 2025. Dr. Li, you may now begin your presentation.
Sure. Okay, thank you. I'm Bingsi Li. First of all, I'd like to express my gratitude for having this opportunity to share our experiences on upgrading and validating liquid biopsy tools with you today. So here is the outline of my talk today. I have 4 parts. I'll start with background, then move to technical highlights and a performance overview, then wrap up with future directions. Let me start with the background. As we know, by analyzing tumor shed components in blood, liquid biopsies provide a new non-invasive approach for comprehensive view of the tumor molecular landscape and a real-time info on tumor evolution. This makes it a valuable tool across the cancer care continuum. First, liquid biopsies captures the molecular information from the entire tumor, thereby overcoming the limitations posed by tumor heterogeneity. Second, liquid biopsies can be done in a series, which means it offers a real-time video of tumor evolution rather than static snapshots. Third, multiple biomarkers provide multidimensional signatures, which means a more comprehensive analysis. Lastly, blood testing is much less intrusive compared to tissue surgery or needle biopsies. As I just told you, the advantages of using a ctDNA assay. Now let's consider the limitations. First and foremost, sensitivity is a significant concern. The majority of DNA molecules in circulation originate from normal white blood cells, meaning that tumor-shed DNA molecules is a very small portion of total. Identifying these rare tumor DNA molecules is like to finding a needle in a haystack. Tumor burden, which is a proportion of ctDNA in blood, can change dynamically throughout a patient's disease course. So it is essential for medical professionals and biopharma researchers to understand how the test performs under different conditions so they can leverage the strengths while mitigate the weakness of different assays. Apart from tumor burden, what are other factors that impact the detection ability of a ctDNA assay? And what are the relationships among these factors? These probably are the first thoughts came to a tech scientist when she or he is about to tear apart and upgrade an assay. Previous studies have shown that the probability of observing a single tumor variant in cfDNA can be approximated by a Poisson distribution. So if we define detection as observing more than one ctDNA molecule, it can be written as 1 minus Poisson of lambda. When multiple variants are considered, the probability can be calculated as 1 minus e to the power of negative n times f times k. So what have we learned from this equation? Let's break it down step by step. First, if you want to develop a sensitive assay, you can increase n. This means increasing the blood volume, the sequencing depth, or the adapter tagging efficiency, or all. I'll dive deeper into these factors in later slides. Second, can you increase F? Unfortunately, no, because this is an intrinsic feature of tumor shedding. Different cancer types differ dramatically, and even within the same type of cancer, F can change dynamically, as we just demonstrated in the previous slide. Third, you can adjust the K, which involves using larger panels with more recurrent mutations. However, it is important to note that cancer-related mutations are usually sporadic in the genome, so there's a practical limit to how much you can increase K. Lastly, what have we learned from the relationship between these variables? N, F, and K are all equally important, right? This helps to prioritize your tasks if you're the tech lead, but never overlook the underlying assumption. Background errors from sequencers, PCRs, or even fixatives in the blood collection tube, all these technical factors can contribute to the background noise. So we know the theory, now I'll move to the technical highlights. The Burning Rock tech team started developing ctDNA assays almost from 10 years ago, and in 2021, the BNR's OncoCompass assay was showcased in Nature Biotechnology, where it demonstrated exceptional performance. Following this, a standard version designed for lung cancer was developed and submitted for regulatory approval in China. During 2023 to 2025, the OncoCompass series underwent a significant upgrade to its ultra-deep version. This enhanced version incorporates unique molecular identifiers technology, an upgraded reagent formulation, a larger panel, and a new data analysis pipeline. This is designed to detect lower frequency somatic variants, including single nucleotide variants, insertion and deletions, fusions, and copy number variations across multiple cancer types. I'll touch base on these points one by one in the following slide. First, let me walk you through how the larger panel was designed. To ensure the panel's clinical relevance, the bioinformaticians, the molecular biologists, and the medical scientists teamed up together. They first conducted a population-level data analysis to identify recurrent mutations across multiple cancer types. Then they investigated the patient-level data in detail to optimize the panel design and the targeting method. And finally, clinical guidelines and clinical trials were reviewed thoroughly to support both the current medical standard and also well-positioned to support future applications. Now, let me tell you how the molecular biologists improved the reagent formulations. First, they improved the UMI tagging technique. As a result, the library yield is significantly improved. Also, the unique depth, which is a surrogate of N, if you still remember from the detection probability slide, now reached almost like 4,000 on average. Second, the coverage uniformity, especially for the GC-rich regions, also improves dramatically. Thus, the variants located in high GC regions now shows a higher detection rate. Lastly, let's move on to how the bioinformaticians improve the data analysis algorithms. First, several statistical models were applied to collapse the UMI tagged reads into duplexes or families, thus removing pervasive random PCR and sequencing errors. The machine learning models were trained to identify recurrent artifacts such as DNA damage patterns and sequencer-specific error profiles to further reduce the noise. Finally, white blood cells pairing was also provided as an option to filter out clonal hematopoiesis-related errors. With all these steps, the error rate reduced about 10-fold across the entire panel, bringing this to the 10 to the minus 7th. Furthermore, the test reproducibility has also been significantly enhanced. For example, the 0.2% reference standards were repeatedly tested here, and the color indicates the positive agreement rate with the ground truth. As shown here, the new pipeline has significantly improved the detection stability compared to the older ones, especially for certain variants. So far, I've told you the factors that affect the detection ability of a ctDNA assay and how we made a comprehensive upgrade. Now let's move to the validation part. How do we evaluate and validate the upgraded performance? Following guidelines from regulatory agencies and white papers from professional organizations, we conducted rigorous analytic studies with over 2,000 tests. I will not go into the details of the validation process. Instead, I'd like to share some tips when doing this analysis. For example, when doing the LOB study, I'd like to point out that the age-matched healthy donors is critical, as clonal hematopoiesis is age-related, so it is recommended to evaluate the assay with age-matched donors for objective assessment. For LOD studies, it is well recognized that LOD is highly correlated with cfDNA input amount. So when choosing or comparing tests, it is important to specifically link the LOD with the input amount. The accuracy study and the precision study can be combined, analyzed all together. It is important to notify that it should be done with the lowest input amount, and also the samples should be prepared targeting 1 to 1.5x of the established LOD values to offer a thorough investigation of the borderline samples. Okay, now let's move to the future directions. First, improved performance usually offers opportunities for new applications. As we discussed earlier, the UMI condensing helps correct amplification bias efficiently. And here is the without UMI correction, and the upper panel is The upper panel is a standard version without UMI correction, and the lower panel shows the ultra-deep version with UMI correction. As you can see, the baseline is much tighter. And this is a cell line spiking experiment targeting 2.7 copies of these 3 amplifications. And you can see they popped out in this with UMI correction while missed without it. This is particularly helpful for detecting copy loss in synthetic lethality genes, for example, MTAP loss. For example, in MTAP-depleted tumors, it can be targeted using PRMT5 inhibitors, with some drugs already showing promising results in early trials. And this showing CMV detection plasma samples from lung cancer patients with or without MTAP loss. Now let's turn our attention to other biomarkers that can be found in the tubal blood. In addition to genetic variants, epigenetic alterations and other types of biomarkers have garnered significant interest in recent years, and this is largely because they pose certain characteristics that are not present in genetic mutations. And let's hope with more effort in the field, we will open more doors for new applications and new technologies. As we know, technology advancements, clinical validations, and regulatory approvals for progressing are usually progressed hand in hand. And today, my talk has primarily been focused on how we enhanced a tumor-naive mutation-based assay. And this assay is mainly used for patient selection, molecular response, and relapse monitoring. However, we're also excited about the emerging applications that lie ahead. We look forward to more breakthroughs and evidence accumulated in collaboration with all of our colleagues and partners. Thank you all for joining me today, and I would be happy to take any questions.
Thank you, Dr. Lee. We do have some questions regarding your presentation here, so I'll go ahead and start with the first one. Our first question asks, how is the concordance of this assay across different labs?
Hey, that's a good one. So we have just finished our concordance study between our Shanghai lab and the Guangzhou lab, and the results are quite satisfactory. And right now we're in the process of transferring this assay to our US company, and we're looking forward to the feedback from our US colleagues.
Excellent, thank you. Our next question here asks, since learning the error profile is critical, do different sequencing instruments matter?
Yes, a very good one. Actually, we tested different models from the same manufacturer, also across different manufacturers. Actually, the error profiles can be quite different. So the bioinformatic pipelines have to be adjusted, and different versions, we recorded different versions, and they have to be applied if you change the sequencing instruments.
Okay, good to know. We have one more question here for you. This question asks, besides blood, can this assay be used with other bodily fluids such as urine or cerebrospinal fluid?
Yes, actually, it's a very good one. We have tested this with urine and submitted a poster in last year's ESMO Asia conference, and for cerebrospinal fluid. Sorry, I'm not an MD. The standard version has been in the standard version has been used on that, and our colleagues just published that on Nature Medicine, I believe 2 months ago. Yes. But the ultra deep version is still being developed. Yes.
Okay, wonderful. Thank you, Dr. Lee. Thank you again, both for your time today and your important research. We would also like to thank LabRoots and our sponsor, Burning Rock DX, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.